progesterone. So the other important aspect of the ovary is that it ages faster than any other organ in the body. So in my lab, we don't study ovarian aging, but the developmental principles that we're learning about the ovary can actually inform about the process of ovarian aging. And so this is essentially a graph showing how organs in the body age, and you can see that the ovary essentially becomes non-functional in women at around 50, loss of the germline, no longer produces hormones because of loss of those follicles, compared to other organs in the body which age much more slowly. And so a new field has emerged in ovarian biology starting to think about this concept of ovarian aging and how to slow the process of ovarian aging down so that the age of the ovary can be more consistent with the age of a human being. The aging of the ovary is important and consequential. So when the ovary ages, this leads to different susceptibilities to disease. So for example, after the ovaries stop functioning, and whether it be over the age of 50, which is normal menopause, or for those individuals who suffer from early menopause, this leads to to challenges with the cardiovascular system, changes in the immune response, more brittle bone, so osteoporosis is increased in women whose ovaries have stopped functioning, as well as cognitive decline due to lower levels of estrogen. So thinking about how the ovary forms, thinking about the ovary as an endocrine organ, not just as a reproductive organ, is really important for overall health and human health. The other important aspect to think about the ovary and ovarian function is how society is changing in terms of family structure, as well as when individuals are choosing to start their families. And this is a graph from the Centers for Disease Control looking at birth rates. So this is from the point of view of women, but looking at birth rates of first child in the 1990s compared to 2019. So in the 1990s, most women were starting their families in their 20s. So you can see here that this is the largest group of when women are having their first child. 30 years later, we can appreciate that this is changing tremendously. In fact, the fastest group that are having their first child, the fastest growing, or the largest change, I should say, is in women who are in their 40s to 44 age group. So now at this point, we are hitting up against the loss of that ovarian reserve, the loss of follicles, the loss of hormonal support, the capacity to be able to carry a baby to term successfully, but also the loss of the germline, which is essential to have a biological child. And we can see that the two other fast-growing groups that are changing from 1990 to 2019 are women in the 30 to 40-year age group. And I certainly am one of those. I had my first baby at 35. I had my second baby at 40. And so this is very consistent, particularly with women who are delaying childbearing so that they can have careers as well as have a family. What this means is that there is increasing use and access to the assisted reproductive technologies, because having a baby at 35, at 40, at 45 can be a challenge. And so therefore medical support is required for some women to be able to do this. The other aspect of thinking about fertility, of ovarian biology, of reproductive biology, is that there is a significant inequity in individuals who suffer from these diseases, but also in their ability to access reproductive care. And so this is something that my lab also thinks a lot about now as we're building models of the ovary, and we're thinking about how these models can be used to understand development and disease, is to build models that are more representative of a diverse population and not just studying ovarian biology, oocyte biology, reproductive biology in one race and one ethnicity. So in the United States, Black women are nearly twice as likely to experience infertility than their non-white Hispanic counterparts. And Asian women in the United States are least likely to access assisted reproductive technologies early, meaning as they're delaying their childbearing, this is again leading to problems with their fertility because of the ovarian clock. So how can we reduce the burden of ovarian disease and reduce the burden of infertility? So the most important thing is to generate new knowledge on reproductive organs. And in our case, we build new knowledge on mammalian ovarian biology, understanding the cell and molecular regulation of how those primordial follicles are built and how those follicles are activated in the process of follicular genesis. Most of the work in ovarian biology is performed in the mouse as a model, and with very little ability to study human cells. And that's because access to human reproductive tissues, in particular access to human ovaries containing follicles, is very rare. The only way to access these tissues is if women are seeking assisted reproduction and preserving ovarian tissue to be used later after cancer therapy or chemotherapy agents. And those tissues are very difficult to come by research. So generating human ovarian models using human cells would be a very important model for the reproductive science community. As we're thinking about toxicology, there's a lot of evidence to suggest that the oocyte and the ovary are exquisitely sensitive to environmental agents, that we need models to be able to screen environmental agents for their effects on the ovary and effects on the germline. And so we believe that pluripotent stem cells can create these in vitro platforms that are amenable to screening. Understanding environmental impacts on the ovary and the germline means that we need to be able to study the ovary and the germline in utero. So how is the ovary created during embryonic life, during gestation? How does the ovary change during puberty? Because this is when you move from the activation of those follicles and the degeneration in the center of the ovary to the ability of those follicles to become ovulated. And then also in the reproductive years, so access to tissues with follicles. When I first started working in the stem cell field, this was at UCSF with Renee Riopera. So when I was at Baylor College of Medicine, I went there because I wanted to learn mouse genetics. I wanted to make